Okay, we're on record?
Oh,
Alright.
we weren't recording
So,
before?
yeah, so let's go over this again. You need your microscope, slides, cover slips, eyedroppers, drawing pencil, onion, which I'm going to cut up in a bit, toothpick, methylene blue stain, which is that little bottle. You need a scalpel. Now, let me emphasize, be extremely careful with your scalpels, because scalpels are very sharp, and if you touch the blade, it can very well cut you. That's kind of what they do. So please be careful. I can show you how to use them. I may be the only one that uses it today. We'll see, because I can cut up the onion for you. And then also we will need a cup with some water.
Where did the rubber thing go?
That is a scalpel, yes.
Do we have the thing to clean off?
You know what? Let me see
That is
here.
not a scope.
That is a scalpel.
That is not.
The cutty thing.
Mr. Belcher, this is clearly a scalpel, correct?
Yes. Be very careful.
Later,
Excuse
Okay,
me,
you
I
can wash these out and use
apologize.
these as your cup for water.
All we need is our
Oh,
onion
we're
because we
recording,
got
sorry.
this.
Not it.
How do you turn on water?
This one.
Alright,
That's gas.
No, that's gas.
They're
It
both
says
gas.
gas.
They're both gas.
I need a little bit more than that, but not like too much. No, we're not doing that. We're not doing that.
So what I'm going to do is I'm actually going to cut up the onion and just hand everybody samples.
This is our professional.
Which one? Wait, which one did you
Alright, that's good. Just try it.
Which way?
Not that way.
That's what I was asking.
I don't know.
Against the rules.
Here.
They get to see us fail on
We're
camera.
not gonna fail.
Wasn't it
Good.
knob we're supposed to turn?
Yeah, this one.
What?
The middle one.
We did and it came out white.
In the what?
This one.
Oh,
Not.
it's just too pressurized. Oh. That's
Alright.
Yeah, maybe use the other. Hey guys, the other
I'm going to
one.
cut off a sample
The
for all the
other
groups,
one.
and we'll start with just
The
one.
other one?
Yeah.
That doesn't
Hold
look
on,
right.
hold on, wait.
Don't do it, don't do it.
No.
Don't do it.
Oh,
Let's not just go
That's
through
a
it.
horrible.
Wait, which one have you guys been using?
This one.
It's just,
But it's white.
it just says, it's just pressurized. It's fine.
You think so?
Just leave it, because the water out there is too. So leave it and it will be pressurized.
I wouldn't drink that.
This was
Well, we're not gonna drink it.
Well, that's Mr. Belcher.
No, it's coming back. It's coming back. It's coming back.
We'll just, here. This look,
It's clearing up. It's
What
clearing.
are you doing?
I'll get some from over here. That's like really clear.
I'm very curious to see it now.
I've actually eaten a straight onion before. I thought we
Turn to the left.
And now,
That looks bad.
it'll get clear like that.
Luckily, they shut down.
We need our onion.
He's cutting it right now.
Yeah. I took a bite of my ice cream and
Seth,
there was an onion in
Tell your
there.
best joke to the camera.
It wasn't very.
No,
You have no good jokes?
no.
You can use scissors. I'll tell you what, if you've got scissors or a thing, you can use that. Basically, you're going to cuff a small piece and prepare a wet mount slide with that, just like we did last week.
I need
Oh, the membrane? Yeah.
Here.
Actually, you know what? Just to make it
That might
easy,
be camera.
that might be enough. Yeah.
Will there's a water bottle flip on the camera?
Scissors here.
Try to use
I
your
would
forceps
try.
for these parts.
Not on camera.
I can do.
Okay. Get that with your forceps. We're going to cut these down to size a
We
little
need
bit.
forceps.
Yes, please.
Seth,
that's
you were
the
supposed
onion.
to
I
be
was wondering
prepared.
what
You had
This is the
one
film of the
job.
onion that we're going to use to study the wall.
Don't squish it.
Where do we put it
Carrying
on?
a scalpel.
Seven. I see.
Alright, forceps.
We need to like cut it
I
in
am
half.
not gonna drop this though.
Cut it in half.
We're going
No,
to cut
we're
these
not gonna
down to
cut it
size. You're basically going to cut just a tiny
And then we
sliver.
do a drop
Or
of
that.
this,
Use
right?
this.
Wait until you set this.
Look, we have our whip now.
Water, yeah,
Cut
I do it
just a
straight
Where's the
section of this
dropper?
off and then try to lay it out
Do you have
flat
the
on a
cover
slide and prepare
Yes,
a wet
they
mount
are
slide,
right
just
here.
like we did last time.
But
Okay, we need to prepare wet mount slide.
Yep.
do
Just
you
like
put the thing on
So
or
remember,
you
for the
put the
first
water
wet mount
first?
slide, what you're going to do is take your
Water first.
slide, you're going to put a drip of water on it, you're going to lay as flat
Just a
as
drip.
possible, you're going to lay a
Dude, no,
thin
that way we-
just
In front of the camera. In front of the
of
camera.
onion
Oh.
piece. That should work.
Well, we didn't cut it up right.
What's better if you do it on a flat surface?
We need to, we'll see.
I know what to do, Seth. Look. Drip of water.
Now, we need a,
Now we need this.
Now we need the
We're
onion.
gonna cut it to
No,
size.
hold on. We need
Okay,
like just
where
a
are we
little
cutting
bit.
it on?
I don't know. We don't have
Just
like
do it
a.
on your lab
there,
manual.
right there. No, not on the lab manual.
Kind of
Okay.
there.
Just cut it. Just like a little piece out.
Wait, are we cutting it vertical or
This
horizontal?
much.
So this one.
It doesn't matter. Just
You
cut
cut for your
it.
first one. I can cut up another
We
piece.
already
Oh,
did.
you got it?
There
You
you
got it
go.
good?
So do we put a drop?
There
Yep,
you
you're
go.
gonna put the.
This is
Well,
the
no, not the blue
dullest
yet. Don't put the
scalpel
blue
ever.
stuff on yet. Don't put the
No,
blue
no, no,
stuff
no,
on,
no.
just
Just
the water.
scrub it with the
We're gonna
forceps.
look
Don't
at
put it
it
in my
with
hand.
just the
Good idea,
water
that's a good
first,
idea.
and then we'll put the blue stuff on it.
Nice.
On another
Okay,
sample.
now we put the thingy on the thingy.
What is that?
On this
I could
thing.
not have put it better myself, Jonathan.
Thank you.
Scientific.
And now we put the cover slip on.
That thingy.
Where's the cover?
What'd you
I
do with
just
the cover
had
slip?
it.
Okay, so let's go through the procedure. Prepare a wet mount of the onion
Yeah,
epidermis
take it
skin
out.
using the following procedures. Remove several layers of the yellow onion, which would go from an inside layer. Cut a small piece of onion, produce a single layer piece about 1/2 by 1/2, which I've sort of done. This piece is called a scale. You're going to take the scale and break it. You're looking for the thin, transparent skin, which I think I pretty much gave y'all anyway. You've already got it. On the inside surface of the onion scale. Sometimes what we'll do is we'll cut off a chunk of onion. You just pull the scale off, but in this case it already happened. Use your fingernail to peel up one corner of the epidermis from the scale. Make sure not to crush it. Again, you already have it, so you don't need to do this. Prepare a wet mount of the thin membrane, which you should know to do. Remember, put the slide down, then you're going to put a drip of water on the slide, put the specimen on top of that, and then put the cover slip on it as carefully as possible. Make sure that the epidermis lies flat and is not bigger than the drop of water on the slide. So it needs to be as thin as possible, laying across. If it wrinkles, try to straighten it with a toothpick or a pencil or pen. Place another drop of water on the epidermis before putting it on the cover slip. So you
Are we
got
not plugged
the
in?
drop of water, then the epidermis, then the drop of water on top of that, and you're gonna push them together.
We have a technical difficulty.
We already put the stuff on.
Why is it not working? It worked before.
I hope that works.
It's not working?
It's on. It
not
says,
working.
dude, the light turned on.
Yeah.
It's on.
Yeah, but this isn't.
It's on. All right, there you go.
Oh, there we go.
Yep.
Okay.
Guys,
Hit me on.
guys, we did it wrong.
Yep.
We did it wrong.
We didn't put this stuff on.
We weren't.
He said don't
No,
put
no,
the
no,
blue
don't put the
stuff
blue stuff on yet.
I said not yet.
Just the water.
Oh.
Yes.
Okay.
No, that's the next one.
All right, so here's what we're going to do. Observe the onion cell under medium power, which is the 10 times objective. So turn your, remember to lower the slide if you need to, and then turn the objective to make sure it's at the 10 times mark.
10 times mark.
And then we're going
That's
to answer
25.
questions
No,
1 and
it's
2 in
not.
the
Oh,
results
it's 10. You're
section.
right. You're right.
So, question 1 that you're going to answer is, after looking at the unstained epidermis, describe the general shape of one cell. In your own words, write down the
Yeah,
general
we are
shape.
the master of
Really
this.
see it. It's just clear. You want to focus like it? It's just white.
Yeah.
Who wants to? Yeah, it's clear. Like, I don't know. There's a tiny hair. That's all. Is it on time? Yeah. Like it's see-through.
It should be somewhat transparent see-through, but you should be able to still see.
Can you see the
Let's
top?
try
Which is
our,
why we do the blue stuff, so we can
let's
see
try
it, right?
our light off real quick and see if that helps focus it. You might have to bring it up pretty close to see it.
Focused on something.
Oh, yeah, like that.
Wait, did you find
You
it?
mind if I
It's
try?
focused on something.
I got something, but I don't think
Does
it's.
it do anything? Okay. Do you see a difference?
I see
Well, I think I see something.
You know when I have the light and don't have the light.
Yeah, I think it needs.
That's not. It's not.
Let me try.
There's something there. What was the flavor of an onion? I don't know. Like after it's cooked.
Oh, okay, wait, hold on. We do have something.
Trust me. I think
The reason is that the layer is not.
did it?
Got it.
Was it either?
You got it?
Was either cheese
Uh-huh.
or the bubbles?
I got it earlier.
Oh, that was
I got both. And I took a piece. And I thought it was either cheese or potato.
just a little bit there. Wow, that looks amazing.
At least it wasn't in your
Delicious.
Delicious. There you
I
go.
put peanuts in my
So we got bubbles and
another time the
There we go.
restaurant
With
dropped
bubbles?
a nut.
Ah, I got it. We'll see. It's
Not bad.
Center, but if you look through there, I got it. We can adjust it as needed. We
got it.
You got it?
Oh, there it is.
Yeah.
Okay. Okay.
Ooh,
Like
let me
very
see.
at the tippy top. Okay. You're like, I am the light top.
What's that?
I'm just gonna check it real quick, just to make sure. Oh yeah, that's good. Well, actually, you know, hold up 1 second. No, it's not that. It's we may be looking at a water pocket.
That's what I thought.
That's
So I was
what I
trying
was
to fix
thinking.
it.
There, oh, there we go. There it is. There it is. Okay, now if you look much closer at it, you'll see the little ridge lines. That's what you're looking at. The big lines that you're seeing is just water pocket, and then the little lines are the actual cell. They should all look kind of in sync.
Oh yeah, I got. Oh yeah.
See little lines.
That makes more
Look at
sense.
the water. I mean, not
Yeah, that's what I was talking about.
Yeah.
They're just gonna watch us look
You're
at it.
you're looking right at it.
Really?
Yeah, they're just gonna
But
watch
it's
us stare
hard
at
to tell.
something.
Like, I'm
and then
done,
they
right?
go,
I can see them, but you can't see it because the slide is so bad.
At least give them some
Let me turn the
comical
light right light
relief.
on, see if that helps.
comic
What?
comedic relief.
No.
Like,
Start telling
I'm hoping this
scientific
helps.
jokes.
But we'll see.
Scientific jokes, nerdy
really-
jokes. Okay, hold on.
This is a little better. This is, you are actually looking
That's
at
kind of cool.
it, but, yep.
Yeah, we're not gonna see anything inside the.
So bad.
Put the phone in your face and not.
today's the
Good
23rd.
try. It might be blocked by this.
Yes.
Yeah.
Oh, no, that works way better. Good job, man. Let's put that. Yep, that's way better.
Oh, he gets a good
The
job.
line underneath is perfect.
so-
Try that now and you'll see the difference. You'll see
Wait.
it very clearly.
Like,
Oh yeah.
There's their light doesn't work. Ours does.
right, exactly.
Alright,
Dude,
so let's
ouch,
look at
that
our
hurts
lab
my
manuals
eyes.
again, wherever
Does
it's
it?
going somewhere.
Dude,
Look.
don't make me go like instantly blind. Oh wow, it's like
Yeah,
it's not. No,
if
it
you need
doesn't
to
hurt.
shine your phone light underneath
Wait,
the
let me see it.
lens, you might actually get
Hold
a
on,
much
look
better
at
It's
picture.
just like shiny. It
It's
makes it
literally
shiny.
blinding.
That was Will, he
Before?
discovered that. We're gonna give him
Thank
that.
you, Will.
Thank you, Will.
credit.
Oh, I kind of
It's like, yeah. You know?
So,
Kind of looks like a
Will coming in clutch.
rubber silver type. I don't know.
It looks like
mixed in
onion.
like a brick wall.
You know? It kind of looks like onion.
That's yours. I think-
What?
Wow.
No
It
way.
does. It's crazy.
Crazy right now.
How to pass the time.
That's yours.
That was fun.
That's yours.
Oh, wait, no, this one's yours.
That's mine? Okay,
Wait.
that's yours. Somebody's?
You-
And this thing is
no,
empty.
This
I think
is
that might
yours.
be mine.
This is.
Yep, aha, that's why I was missing it.
Oh,
Okay,
nice.
so, after looking at the unstained onion epidermis, describe the general shape of one cell. So try to identify the cell with the cell wall and try to describe the shape in number 1 of your lab manual. Then try to describe what you see inside the cell. That's going to be the first 2 questions here.
Wait, how do we
It
know
reminds me
which
of, like,
part's the cell?
The cell are the little individual, like the cell you're looking at is the individual pockets, like that are lined by the cell walls.
So the lines
Maybe if I
themselves?
get an air
The lines themselves are the cell walls. The actual circles that you're seeing are the cells, the individual cells. We're gonna go ahead and look at them again.
So the circle
Yep.
looks like a circle.
It reminds me of
Picture again on his laptop.
I said it looks like onion.
I've got my drawing of it. It's just faint line.
It actually smells like onions in here now.
It looks like an onion's liver, but 10 times as big.
An onion's liver.
Okay, hold on one second. I'm gonna put a picture back up of
Onions don't have liver.
the cell that you should be seeing. You may not be seeing it quite this close. It's probably a slightly lower magnification.
Should we move the camera to the...
No. You're fine.
Can't see that.
You don't get to see. Have to purchase VIP to be able to see that. It's only $9.99, guys. Pay it to me. I'll take it.
That explains the meaning of the blue
Jonathan,
one.
how
It's
tall
just
exactly
weird,
are
but
you?
the blue one-
I don't know.
Good conversation.
Okay, so you should be seeing something like
6'2".
this. Okay, and you
That's
got
what
your
I
cells
laughed.
right here.
That was a
These
couple
are the
months ago.
cells, and then these are the cell walls that are like boundary it.
Yeah, what we see is like very faint newspaper lines. And we're trying
We're-
to
You're doing it
I
wrong.
Like
couldn't
that.
find it. Ah, there.
It might. This slide is a little bit off. I think.
Okay, I
Okay.
got it. I can't get in.
No.
I see bubbles, but I'm not.
This is mine now.
Yeah, that's about as clear as you can get. There's a reason we're going to cover these in blue in a bit. You're going to see them even clearer. So for now, what it may look like is that they're almost
just
invisible
like-
or very transparent and clear. That's
Okay, that's
okay.
what I'm right.
What?
Okay, now we're going to move on. Okay, so once you're done looking through that, we're going to prepare a new slide.
Alright.
Go back to our thing here. Yep, so now we're going to prepare a stained wet mount using the following procedures. Follow the same procedures originally that you did to produce a wet slide for the onion, but do not put the cover slip on.
We're gonna just like leave this here and
Yep.
get a new one. Just make it entire.
Bring in a new slide?
Yep, so prepare a wet mount just like you normally would with your we'll get a new sample, and I'll cut it out for you.
New sample. He's got it on. Oh.
I messed it up.
Not already.
I guess we
I
can
mean
throw it as
already.
onion sample.
It's too much water came out of it.
Are we gonna put the drop of water on that?
We are, but we're gonna not put the drop of water on top of the specimen yet. We're gonna put the drop of water on the bottom, lay the specimen on the drop of water, and then don't put the cover slip on yet. I'm gonna try to see if I can. Aha! There we go. This is much better.
Is that too small of a drop?
No.
No, you might want to add a little bit more, just a little.
Why'd you say no then?
Hey, watch out, watch out.
You're gonna mess
Wait,
it up.
hold, in front of the camera, in front of the camera.
You're gonna totally. Yeah, so you can film your demise. Oh wow!
Thank you. That was perfect.
You stroke of luck. You're like, and
That
then
was
dropped it.
perfect. Precision.
And that was the
He
opposite
said
of
something
precision.
about water.
Yep. Hold on. Be right with you. I'm just gonna try to get y'all's your new samples.
We might need a thinner one than that.
Yep. Don't worry. Don't worry. Here's sample number 1, and we'll lay it. You can use your forceps, and you can cut it to the size you need.
You want that?
It smells like onion.
Oddly enough,
Wow.
when you use an onion,
Great
That was
observation.
profound, Jackson.
Scientific discovery.
this is not coming off the way I wanted it to.
Right?
That looks so smooth.
What we're looking for here is the skin
You know how
that
it
comes off this
Gives like
outer
profound
peel.
quotes every time you open Grade Link.
No, what? You're not.
It doesn't?
No, I don't read them.
Oh, well, you know they do.
I didn't know that.
Next one, Jackson Spread like onions smell like onions.
Yeah, but that's actually not a bad idea. Try to pinch that little membrane.
They quoted Kermit the Frog, dude. You should
I'm
read
allowed
these.
to, yeah.
Well, I'm saying because you don't finish it.
I usually
Almost.
finish it, but I forgot about it during that class last time, and I think it was folded by then.
Oh, we are.
I was done. We'll use this one. Use that, yeah. When I went to my flashlight, I have
Yep,
this.
almost that.
And so
Pull
it
up.
just shows me pictures. And and this
There we go.
I can make it do that. Well, I
So here's what
did
I'm
make
gonna
it
do.
do that
I'm
on
just
my
gonna pull the thing
old tablet.
So here's
It
what's
makes the tablet.
things not covered in onion.
It's a new update for my phone. I've never been able to do that before.
Oh yeah, one of my friends.
Okay, and you can cut whatever size you need out of that.
It's,
Alright.
this is.
Wait,
Zach.
where's Sam?
Seth.
That I know of. It's an iPhone 6S.
This should be
Mr. Motor
whatever size you need out of that. You might want to flatten that out a bit more.
Every time there's a new iPhone, like 3 minutes later, there's another.
Okay, now, let me read through the instructions now that you have your membrane. So you're going to prepare the slide, lay the drop of water on the slide first, and then lay a small piece of the membrane on the drop of water.
Try to lay it flat.
We'll start with that. Once you've got that, move on to our next step, and this is where it's going to be a little tricky.
Okay, that's good. I'll move it. Now what?
Once you've layered the epidermis on it, now listen up, listen very carefully. You are going to take one drop, only one, of the methylene blue stain, and you're going to drip it onto the epidermisil and then put on the cover slip.
Alright,
So you're going to put it
Where's
on
the
the
cover slip?
specimen
Wait, no,
and
no, wait.
put it on the
What-
cover slip. Try to make sure
No,
you're
we're
getting
gonna put this
exact
on
fill
next?
as best as possible.
Okay.
And only one
Moment of
drip.
truth.
Trust me, it is really blue.
Yeah, it's like blue food coloring. It's going to very, very
Okay, good,
strong.
it comes up. We have a big bottle of
Now,
it.
after this, we need to let the slide sit for 3 minutes.
Nice,
Okay,
nice,
so we're
nice.
just
I
gonna
was laughing.
chat for a bit while we let it. Once you've got the drip
Why is
on there
that?
and you got the cover slip on there, and make sure it's all flattened out as best as possible, and let it sit for a while.
Bro.
Wait, we put the cover slip
Where's the
on
cover?
now?
No, no, no. We'll
Once
get
you've got the
that
blue
fine.
drip on, you lay the cover slip on top very carefully. Make sure it's in the center.
Where's the ta-da! Science. Okay.
They're pro-
Oh, that was bad.
Push it down. You gotta push it down. No, not with your finger. Not with your finger.
Let me see it. No, no, no. Let me see it.
Okay, fine.
Oh, whoops.
60,000 cameras on there.
Okay.
There you go. Good.
Nicely done.
For now, just
Give it
let it
this
sit
up.
for a little while. If you want to chat for a couple minutes.
Yeah, let's chat for a couple minutes.
How's it going, guys?
Supposed to hold it there.
What? For a couple minutes? We'll leave it here.
If you want to see something, just
Probably will edit
my
some.
brother and his best friend just being
That's
good.
good.
We were making a show
There's a lot
called
of
Tweedledee
things.
Tweedledum.
That's
It's
just
about them
time.
two.
That's okay.
And this is the first.
Especially talking about Jonathan's height and his scientific jokes.
I plan on leaving those in. That's what makes that fun.
So I said I was 6'2", but some kid came up to me. Yeah, whatever. But he was like, yeah, I'm 6'3". And
Who's
he's
like
like up
that?
to here, aren't they?
Who?
So, Dalton.
And they,
Okay.
it
Don't-
just keeps
So you're like
them
6'5" or something?
What?
And they're
So what, you're like
speaking-
6'5"?
and they're talking
No,
about
probably like
babies.
6'3".
They're just talking.
Did you say
about
me?
It's a
You're
six-
36.
I don't want to get any
the
taller.
onion?
What's wrong?
Yeah, it is.
Why not?
What?
I want
Ah, reasons.
Well, being
I don't want to
6'2"
stay
is
here.
already cool.
Well, yeah, but I don't want to get any taller.
You're like
You
the
know,
height
if-
of Batman.
If
And
you're
Batman's
so
6'2".
tall, if you're
My
so
parents-
tall, you have to look down at everybody.
Yeah, all the furniture was built
Bruce
into
Wayne
the
is
house.
6'2".
Is he
All
though?
the furniture was built into the house.
We have a
Burn it.
I didn't think he was that tall. Was that our last slide you just dropped?
This
Yeah.
was the
Why'd you
kitchen
put it
and
on like
base.
this?
I didn't. Seth
Right.
did.
Good question.
Dad.
I like the dolphin.
Wait, is it?
Wait, why is there a cup?
Alright,
put
when
it
you're ready, go ahead and
upside
try
down.
to prepare the
Glucose.
slide into your microscope. Try to make sure everything is focused correctly.
Wait,
I
where's
had to
this?
sit for
Dude, where's
cameras.
that blue stuff?
It's,
Right here.
by the time you get it all set up, it'll be pretty much the amount of time we need.
Wait, you gotta move it down.
One drop it back.
We're going to view it under the same exact
Only I know what to
size, so you don't
do.
have to switch objectives. Uh-oh, what happened?
Nothing. He's overreacting.
Is he?
Like normal.
That is the question.
Why does it go under that? It's not supposed to go
Okay.
under that like that.
Dude, what on earth?
Dude,
did
what
you
is
do?
going
Stop,
on?
stop,
I
stop.
can see it already.
Just what is going on?
Not even.
It is not supposed
What does this
to do
twisty thing do?
that. Probably twist.
Here, give me that.
Okay.
No, 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 no.
Oh, I got it, I got it, I got it, I got it.
I want to do this. I need to redeem myself. Wait.
Every time you pull the lever, it goes up. Okay.
Whoa, the lever crunk.
That wasn't very funny.
Remember
Okay,
to shine
it
a
wasn't
light, like shine a
supposed
flashlight under this.
Sweet, sweet, that's perfect.
I think that'll do.
Or should we turn the light off?
Should be about
Wait,
there.
wait, wait.
Just look in, look in.
I think
No,
that's
it's just blue.
There,
Oh,
there
wait,
it
here we
is.
go.
Yeah, once you shine the light, that's it. Check it.
Wait, hold on. Let me
Yeah, that, I
do it.
think shining the flashlights underneath the microscope is actually
Do
working
you think
very
turning the
well.
light off will help?
We can try.
Oh, here we go.
If you're a remote student, I hope
Baba
you're
da
scared of
boo-
the dark.
Oh, wait. It was perfect and then it was gone.
Nice.
Here, here.
There's a glitter. Oh, I see
That's
the-
even darker, dude.
Dude, what's going on?
I'm getting it focused. Oh, it's beautiful.
Just two
Yep.
flashlights.
It couldn't be more beautiful. Just look at it. Look at the beauty.
Oh, I'm supposed to be wearing a mask.
Okay, so now you've got a drawing on.
Am I right or am I right?
So first of all, answer question 3 now that you get a chance to see it. What difference does the stain
Look at the
make in
beauty.
viewing the cell? Try to describe it as best as you can, using your obviously writing. Then the second question will be, you should see a dark circular structure in the cell.
Wow.
What is this structure? You should see a dark, tiny circular structure in the cells. See if you can spot them.
I can't
It'll be labeled. You can see what you should be looking at here
Okay, here's my phone.
on the projector screen.
What is the dark circular structure labeled as?
Yeah.
Or is it not?
Nucleus.
Oh, that. Oh, yeah. And what number is that?
You can't see it.
Oh yeah.
You should see the nuclei.
Yeah, the flashlight's gonna make it.
See it?
Oh,
The flashlight under it.
What am I supposed to do? What difference? What our microscope at home is
call
like
911.
it's a mirror
Mhm.
and a flashlight.
No, 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 no,
And you
no,
have to
no,
like
no,
bounce it off
no, no,
at
no,
the
no,
right
no, no,
angle
no,
to get it to
Stop.
shine
Oh, no,
up.
no, no. Okay.
I'll have to look and
It's
see
like
if that's
on
how it
his
is
phone
with these
on
microscopes,
camera.
'cause
What?
these
I
microscopes
think you're
seem
on
to
your
be a
phone
little
on
too
camera.
I
bright
did it
light.
once, cause look.
Maybe because the battery's dying or something.
Yep.
No.
So again, you're
Turn
looking
the
for those
phone
little
on.
dark spots,
Do
those little dark circular structures in the
this.
cells. What are they called? And do you
When
see
I
them?
hit 0, because my password is 0, and now you know, I hit emergency call. When I freak out and I hit back,
Oh, I'm
I
sorry,
hit
yes.
911 right there. I've called them so many times, and they're like,
I love
what's your
how
You
this
see
name?
those little
being
dark spots in
recorded.
your cells?
Oh, no.
You're looking at the nucleus.
Hey, flashlight, need a flashlight.
Okay.
Now, using a
Where's
blue
that?
colored pencil, if you have them, try to make a drawing of the adjoining onion cells under medium power in the circle below. It's on page 23 of
Farther,
your lab
farther,
book.
farther under.
What do you mean farther?
Farther under.
I don't know what you're talking about.
Make sure that you refer to lab 4 on microscope drawings to make sure you have everything that's needed for your drawing. You need to be able to label the cell wall, the nucleus,
Did I
and
do anything?
Yeah,
chloroplasts
it's
if
kinda
you see
cool.
them.
Okay.
Seriously?
Wait, what are the
Oh. Wow.
Chloroplasts should be these tiny little
I'm trying to get that
things
right.
going
No,
around the cell wall.
there you go. There you go.
Basically, these little tiny things
Yeah.
going
Oh.
around. So this is your nucleus and these tiny little divots.
I'm borrowing one of your blue pencils.
Yeah.
These little, they should almost look like green little dots here on the screen that I can tell, but you're looking for those even smaller than the nucleus, tiny little.
Oh, I see.
Digits.
So it's like specks. or-
Yeah, like
okay.
specks. A little
So
bit smaller.
Why is it so dark?
Oh, do you need some other coffee?
It reminds me of SpongeBob's house if it was blue. That's interesting.
Once you're done doing all those questions, we'll then make a high power resolution, so we'll actually look even closer at it. Oh, I see you're already good.
Hey.
Alright, so.
Yeah, if you've already filled in those, you can move on to the higher power. Yes.
Could I try it on there?
I think 40 is the highest
Just
one.
do.
Oh, yeah, go ahead. Try on the highest power. Go ahead and move it to the highest power if you want to.
Whoa, that's
Be
way too
careful.
close.
Make sure that
Oh.
before you switch it, that you bring the base down and then switch the objectives and bring the base back up.
Oh, now.
That way you avoid scratching
You were
the specimen.
So it's fine. Don't worry about it.
Hey, I found the last one. Let me do this.
how do you spell
Okay. NUC, I don't know.
Oh, I
So
see.
yeah.
You have to have
I like your drawing.
It's very nice.
But
Do you like anything my drawing or I don't remember?
I think
Oh, sure.
I.
Oh, it's actually, yeah, that's the best. Because mine's the best.
A blue line.
No, yes.
Mr. Belcher, for plural of nucleus, it's nuclei, correct?
Dude, what
this
is
I think it's just
Yes, I
nucleuses.
Whoa, dude, look how close you
nuclei
are.
would be NUCL.
Okay,
You,
okay.
you risky, bro.
Watch out.
Dude, that's the only, dude. No, 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 no. Come on, stop.
Latin for
Pictures. Did you
one.
want to look
Look, I'm gonna shine the light.
Yeah, maybe.
Yeah,
Let's see what our
Maybe we should go
my
down.
old, Eng-
For one thing, I bet.
my old English curriculum would make me do the foreign plurals and stuff.
Okay.
So,
Whoa.
Wow.
and
Did you
I
find
can-
it?
Stop, let me do it,
Okay. Oh,
if you want to grab my phone and stable it, I'm trying to draw.
you're not even under the prompter.
Whoa.
arms look extremely hairier on camera.
Thank you.
What?
Yeah.
Now, what did Seth do with my colored pencil?
I'll put it back.
Heard this and this.
And how do you draw an arrow
Wanna see?
to a tiny dot?
Yeah.
I'm just gonna move this around.
Oh! Oh!
That's a blue one, right?
What does it look like? Oh! It's like the hair, but there's a cell in it.
You might wanna look.
Kind of like the hair was.
You have to go around me or this
Oh yeah, it kind of does look like
It's
how the hair
focused
is.
in on one of those lines. Like really
Now we
focused.
only have about
Very
15
well,
minutes.
It's blurry. Oh, there
Quick.
it is. Hold on, can I see my?
Don't-
You might
hey,
wanna
hey,
I'm trying to turn your flashlight
You
on.
can- you can't. With that,
I-
oh. Whoa, you
It's that
can
easy.
do that? And he didn't even put the pets in.
Oh my god.
He didn't even know what he was doing with his own phone. Oh yeah, that was 10 times.
You might wanna look. If you
Yeah,
can't see it
this
way
one
up
was the
close.
highest. I don't know what the highest
What is that,
was.
40?
Mr. Belton, you might wanna look at it that way.
Hey,
It-
hey.
I got it really big.
Seth,
Seth, what's the highest?
you know what my background was? It wasn't actually on my background. You know who it was?
The nuclei.
Oh.
I know that.
Guess.
Michael Jackson.
Do you know what album it was?
I'm turning off
Never mind.
this.
I'm missing a colored pencil.
What's this one?
Seth.
I put it back.
I'm joking.
Do you want to copy?
Wait, which is
I'm going to
do
40
it.
times?
I
Yeah.
find it often easier to just. I don't know if there's like a bunch of.
Oh, wow.
I need a colored pencil.
Not exactly sure what we're looking at though.
I need another view before I draw this.
Exactly.
I hope that they get more samples or something. We can look at those like, yeah, the right thing.
Easy. Alright.
Easy. You better use your manners, young man.
Now, it may be hard to tell with the dark blue of the slide, but what you should hopefully see is some very, very tiny green specimens. Just very, very tiny green specks. You may not be able to see it because they're pretty dark. If you can't, generally what you're looking for is what is called the chloroplasts. They're tiny green specks.
See specks, but they aren't green because
They
we
may
dyed
be
them black.
dyed because of the blue. Typically, they take on a green hue. There's a reason why most plants are green.
Is there going to be a sample lab on Great Big Basin?
A what? A sample lab?
Look,
I'll have
we
to
can travel
look
around the
up
map
for it.
if we do
I
that.
didn't get one yet, but I'll check and see.
My dad will have it on for like.
Oh, false alarm.
You
Those
may actually
specs are
use the same
actually on the
format
lens.
that you did for lab 3 because you're still going to do the drawing, so it's the same format.
The specs are on the
We'll check.
lens. What do you mean? The specs that were supposed to be the little green specs.
Okay, so try to
on the
look. So on the
lens.
medium power,
Look at-
label the
no,
cell
no,
wall
no,
nucleus and
look at
chloroplast to the best of your ability, and then on the high power, try to label the cell
Alright,
wall
so
nucleus
look
chloroplast.
at it and
Remember to
then
draw
turn this
examples
one.
of both.
A little bit. See how it moves around, but it takes the dots with them, so the dots aren't actually on the thing. It's on the. Dude, that's.
I don't think that means what
Yeah.
you think it means.
No, that's because this is. I know. That's what I'm trying to say.
What are we talking about?
Jonathan. What do you mean? The Microsoft itself is dirty.
like
That's what
that.
I was trying to say. Yeah.
But you
Okay.
said the green dots.
I'm going to help you answer a few questions just because we haven't really gone over them. So when it asks you on 7A, what are the color chloroplasts, you're going to answer green, typically. When it also asks you what important chemical do chloroplasts contain, can anyone answer that?
Chlorophyll.
Yes, very good, chlorophyll.
Yay.
Who names these?
Scientific people.
Name Phil.
Why is there chloroplast?
What?
Scientific people named
Phil. There's this lady. Her name's Clara,
Okay,
and her
what
wife
is the
is
function of chlorophyll
her
in a
husband,
plant
Phil.
cell? What does it aid the
Chlorophyll,
plant cell
chlorophyll.
in? Can anyone answer that?
the function of
Yes.
Photosynthesis?
Photosynthesis. There you go. So the function of chlorophyll is to aid the plant cell in photosynthesis. The chloroplasts tend to absorb the water and the oxygen or the air. They move around and help generate the energy.
So. No. Something spicy said.
I'll bring up the chemical formula in just a bit here.
You ruined it.
What happened?
Not even to the beat.
Yeah. It seems like there's a dark.
What are
Ah,
you
missed
doing? Are you trying to? Okay.
Background music for the camera and people
So the
don't.
function of the chlorophyll is to turn the plant green.
Yeah, it's for
And
photosynthesis.
then, I have no idea, guys. I just tried my best. You can probably look it up.
Hey, what is it looking at?
Okay.
It zoomed in on your hand.
That's fine, once we get these.
H2O plus.
That's what I'm probably going.
It's so easy to.
The only good people with that is for math.
You know what I used to do before I
Oh, I messed up.
started oxygen? I would like use the boxes to make colors. Like I
Not
would
bad.
like color in the box. I've hidden mine
No.
in a pocket
Yes.
under things. It's actually
Hmm?
honestly, I thought it was just designed in a jacket.
Okay, now, I'm going to give you one more thing that's very important for the end of your lab. You need to understand what the missing parts to the chemical formula for photosynthesis are. Okay, so H2O is water. So first part of the formula is H2O plus CO2. What is CO2?
Carbon dioxide. Oxygen?
Mm-hmm. I believe CO2 is carbon, then it might be oxygen. Let me check.
No, it's carbon.
Oh, we're taking this mask off. I can't
Wait,
breathe
how
anymore.
is C?
Yeah, it's carbon dioxide.
Mine on my seat.
So what?
Is it like C to the second power? Like
Yep.
that?
CO with a 2 down at the bottom, not 2 at the top. So H2O plus CO2 or water plus carbon dioxide equals
Oh, okay.
C6 H12 O6
Wait,
plus
what?
Wait,
O2.
say that again?
Question?
Yep. C6 H12 O6 plus O2. Now what is O2?
Wait, where do we? I'm so confused. Okay. H.
This is the chemical formula for photosynthesis.
Plus
So,
O6.
water plus carbon dioxide equals C6 H12 O6 all together. So each number is going to be on the lower right of the letter. C6 H12 O6. Yes. You have a question?
C6O.
Nope. C6 H12.
What's that?
I believe it's hydrogen. I could be wrong. Look it up.
I bought this before Corona.
Really?
Yeah.
Why?
Because it was cool.
It is? I mean,
Yep, okay, here we
I
go.
know.
So I found the thing for it. Yeah, I think there was. Alright, so CO is obviously carbon dioxide, CO2. Okay, H2O is water. Now, here's the tricky one. C6, H12, and O6 is glucose.
where are we
So
supposed to
remember that
put
sugary
that?
substance we talked about earlier that was on the lab here? And then O2 is oxygen.
And
Yes.
if you would do
Glucose.
the numbers.
So here's the chemical formula again.
I'm lost. It's the same.
We're writing down the chemical formula right here on number 8 of
Like
page
if you reduce the
24.
6, 12, and 6, it's C2.
So
like
it's
that one.
C6, H2 is not a part of the C6.
No, hold on. Start from the beginning, or from left to right. H2O plus CO2.
That makes sense.
CO2.
Yep, CO2
That's why it says try
That's
to
carbon
balance
dioxide.
the equation. It's the same thing.
equals
Okay.
C6 H12 O6. That's glucose.
I'm so sorry.
That's okay.
So, water plus carbon
Mm-hmm.
equals
Yep.
glucose.
Is this even still recording?
Oxygen, very good. So the English way to say that is
It's for correct
water plus carbon dioxide equals glucose plus
It's for correct
oxygen.
underneath.
I feel bad for Mr. Belcher. He has to go back home and then edit all the footage of us literally just making music with the
We'll
instrument.
figure it out. I may just write
No, it's nice.
It's confusing. It says try to balance the equation.
a penalty for being a remote
We
student.
did
You
not
must
learn
suffer.
this.
All right. With that said, we are pretty much done for today. So I'm going to stop recording.
You might want to put this